So welcome everyone to the second annual Biology Science Slam. It's a chance for the trainees to take over the colloquium, show you what they've got, both in terms of the really interesting research they're doing, but also in some sense, more importantly for the sake of this slam, how well they can communicate that to you. This effort of building the Biology Slam is a group effort, as everything is. The BGSC was a co-sponsor of the Slam this year, together with the communications office in the biology department. And I'm going to call out two very special people who are actually going to take over the mic in a moment, Emma Cowell and Moni Aveo. Those of you who came to the Slam last year may remember them as participants, and Emma actually claimed both the audience choice and the jury prize last year for her talk. And so they've actually done all the heavy lifting for the Slam this year up until today, workshopped the participants, gave them feedback, looked at their slides, gave them feedback. And so there's a lot to thank them for this Slam, and they're going to take it over in a moment. But before they do that, I'd also like to introduce three very special guests we have with us today who are part of the Slam, and that's the jury. Now, why do we have that? Okay, we'll do that. So the jury consists of Ari Daniel, Mary Carmichael, and John Pham. Ari is a senior digital producer at Nova, where he creates interactive games and digital videos about a wide range of science topics. He's also reported on science across 5 continents for a variety of national public radio programs and other outlets. He is co-recipient of the AAAS Kavli Science Journalism Gold Award for his radio stories on glaciers and climate change in Greenland and Iceland. Ari also co-produces the Boston branch of Story Collider, which is a live storytelling show about science. He received his PhD in biological oceanography from MIT and the Woods Hole Oceanographic Institution, and a master's degree in animal behavior from the University of St. Andrews in Scotland. So you could see this wonderful smile on his photo. He smiles like that a lot, and in fact, he won Most Contagious Smile Award in 5th grade. Okay. Next to Ari is Mary Carmichael, who describes herself as a journalist turned catalyst, a storyteller who moved from describing biomedical innovation to driving it. She founded her first venture, a small publishing firm, while majoring in public policy and biomedical anthropology at Duke. After college, Mary joined Newsweek, where she wrote and reported dozens of cover stories and features on biomedicine and health, broke a story that led to the retraction of a paper in Science, and won several national awards. She subsequently moved to the Boston Globe to cover higher education as an investigative reporter, which is when I met her, taught in the MIT graduate program in science writing, which may be when Raleigh met her, and freelanced for The Economist, Scientific American, and Nature, among other venues. In 2011, she completed a Knight Science Journalism Fellowship here at MIT, focusing on human genetics, left journalism to join the Broad Institute at MIT and Harvard, where she helped build and found a family of web portals for securely sharing genomic and clinical data. She then moved into organizational leadership, where she oversaw Broad's academic community of 400+ faculty and 1,300+ trainees. She's currently here at MIT, completing her MBA at MIT Sloan, and is the founder and CEO of a strategic communications consultancy focused on innovation in science and tech. That consultancy, named Quark 4, will launch this fall. And last but certainly not least is John Pham. He is the editor-in-chief of Cell, succeeding Emily Marcus, who was here for the last Science Slam. He's the 4th editor-in-chief in the journal's 45-year-old history, so that's 2 behind me. He joined Cell Press in 2008 as a member of the Molecular Cell editorial team, where he became editor-in-chief in 2012. John Pham has championed transparency, integrity, and author hospitality. Over the past 11 years at Cell Press, he has helped pilot new policies on how data should be uploaded and archived, promoted pre-publication image screening, and experimented with peer review. He's a trained molecular biologist who received his PhD from Northwestern University And during that time, he co-authored a number of papers, including 2 published in Cell. He did postdoctoral work at Harvard Medical School and Brigham and Women's Hospital before moving on to Cell. So I'm going to back up and just talk about the rules and then pass it on. So just as a reminder, the Science Slam, each person will have 3 minutes, 1 slide. It's a spoken word rather than a performance case. So it's no dance, no, as much, and no props. So they have 3 minutes to tell you about their science, and here to introduce both. I'm going to pass this on to Emma and Moni, who will talk to you about one more prize that we're going to give at the end, and then introduce the participants.
Hi guys, thank you so much for coming out today. We're so excited that you're here to come see what our fearless slammers have prepared for you. My name's Emma. I'm a 3rd-year graduate student in the Burge Lab here.
And I'm Moni. I was a graduate student, but now I'm a postdoc here with MIT X Biology.
And we will be your hosts today. So the one thing we want to say before we get started is that in addition to the 3 illustrious judges we have here in the front row, all of you are going to be empowered as judges today. So pay attention during the talks and remember which one is your favorite, because you will be able to vote on your phone or a laptop for your favorite at the end. And this vote has an impact because there will be 2 different winners, the judges winner and the audience winner. And remember, it's not just which project is your favorite, like which science you think is the coolest, it's who do you think sold it the best. So without further ado, let's introduce the slammers. Okay, quickly let's give a round of applause to our 8 slammers. And I'll hand it over to Moni to introduce the first one.
I'm shorter. And before I start, I want to apologize. My intentions are good, but my English is not very good looking, and so if I mispronounce anything, please forgive me. So, with that, our first slammer is Becca Silberman. She grew up in Somers, New York. Yes? Yes. She studied biology at Colgate University, where, fun fact, she studied how energy is stored in antibodies affects their storaging behavior, and so she was like rolling around in dirt, which was a cool fun fact I didn't realize before. And she is now a graduate student in Angelika Amon's lab over in the KI. A cool biological phenomenon she has seen in person, this is so enviable, is glowworms that live on the ceilings of caves in New Zealand. Just how cool is that? So anyhow, without further ado, Becca.
Consider for a moment how many times the cells of your body have had to divide to build your body from the single cell where you started out. And think about how well it's gone, making trillions of cells that function and are recognizably your own. The real drama of cell division comes in at the end, when a cell, having duplicated its genetic information and packaged it up neatly in chromosomes, which are shown in red at the top of the slide, has to then segregate that DNA equally between 2 daughter cells. Equal and accurate chromosome segregation is a challenge shared by organisms ranging from humans to the hardworking yeast that brew our beer, and it is handled in much the same way, with careful regulation and great precision. Of course, I don't study these trillions of success stories. I study what happens when cell division goes cataclysmically wrong, producing cells with the wrong number of chromosomes, which I'll call imbalanced cells. Sorry. So normally, the fate of these imbalanced cells is pretty easy to predict. They either die or survive, but fail to really function or proliferate as well as they should. But there is one context where, in defiance of everything that we understand about cell division and genetics, imbalanced cells thrive, and that is cancer. While imbalanced cells are incredibly rare in healthy human tissues, they make up 90% of tumors, which begs the question, are imbalanced cells more likely to become cancerous, or are only cancer cells able to tolerate having the wrong number of chromosomes? When I first started working on this project as a first-year grad student full of ambition and energy, I collected samples of precancerous growths from the skin and breast and colon and asked a very simple question. Are the cells of these growths, which are not yet cancer cells, but are moving in that direction, balanced or imbalanced? And here I am, a fifth-year grad student, a little more practical and definitely more tired, and I can tell you that the cells of precancerous growths have the right number of chromosomes. Being imbalanced does not make a cell more likely to become a cancer cell. Instead, it is only after a cell has fully transformed into a cancer cell that it's able to tolerate having the wrong number of chromosomes. In my current experiments, I'm digging into this observation to try to understand where this tolerance comes from and what it means for processes like metastasis and resistance to drug treatment. And I'm really excited about these experiments because I'm hoping they'll teach me something not merely about cancer, but about cell division, this ubiquitous and essential challenge, and about the rules that underwrite biology. Thank you.